Okay, hi there. Welcome to this video on DNA structure. So this is all about the structure of DNA right down to nucleotides. So DNA is made of something called nucleotides, and the first thing you need to know is how to draw a nucleotide. So there's always going to be three parts to a nucleotide. It's always going to be made of these three things, which we simplify as these three shapes. So first of all, up the top here, we've got what we call a phosphate. Then in the middle here, we've got our sugar. And then across to the side here, we've got our base, sometimes called, the full name would be a nitrogenous, can't just call it base really, but full name is a nitrogenous organic base. Okay, so phosphate, sugar, base. Now the phosphate is what we call a phosphate group. We don't really need to know anything more about that. The sugar, however, we do need to know something about. So the sugar is what is a 5-carbon sugar. And because it's a 5-carbon sugar, it's called a pentose sugar. So the pent part means 5, like a pentagon has 5 sides. So 5 carbons, it's a pentose sugar. Now there's two options of what the sugar could be. So in DNA, which is the one more common that we have, the sugar is beginning with D, hence why it's called DNA. And that D is deoxyribose. And in RNA, which is another kind of genetic material that other organisms have, it's just ribose. So this R in RNA stands for ribose, the name of the sugar. Now, as the name suggests, they're kind of the same thing, but DNA is deoxy, so it's got an oxygen molecule less in its structure, but you don't need to be able to draw the structures. This is just the level of structure of nucleotide that you need to be able to draw. We've then got our base here. Bases come in two categories, which are called purines and pyrimidines, but actually you don't need those words for your specification. But what you do need to know about the bases is that in DNA, the bases are A, T, C, and G. And in RNA, they're slightly different. So in RNA, they're A, U, C, and G. So we've got a U instead of the T. Okay, so you need to know the four bases in DNA and in RNA. Okay, so if you were asked to draw the nucleotide for RNA and the nucleotide for DNA, the only difference would be the name of the sugar and possibly different bases. If we've got one nucleotide like that, we call that a mononucleotide. So it's the same language as we used in carbohydrates. So we called it a monosaccharide, and then we called it a disaccharide and a polysaccharide. So it's the same here. So you'd call one nucleotide a mononucleotide, two nucleotides a dinucleotide. And then if you've got lots, you could call it a polynucleotide. So the mononucleotide is going to be the phosphate joined to the pentose sugar joined to the base. A dinucleotide is going to be two of these, and the way they join up is they go directly underneath each other like this, and the bond forms here. Alright, so that's the bond joining two together in a dinucleotide, and then if you had a polynucleotide, obviously we're looking at having even more in a line. So I'll just draw 3 for now. We don't want to be doing this all day. Make sure you're getting them off that left-hand corner of that pentagon. And then we've got our bonds in here. Alright, so we've got mononucleotides, dinucleotides, polynucleotides. Now, just like in our work that we've done on carbohydrates and proteins, when we join them together to form these bonds, the bonds are made in condensation reactions. And they're broken in hydrolysis reactions. Okay, so just the same as in the other topics that we've done. Because it's a condensation reaction, it's also going to make water. Don't forget that. So 2 of these joined together to form these, joined with this bond, and it's going to make a molecule of water because it's a condensation reaction. If you wanted to break them apart, you need to add a molecule of water in order to separate the dinucleotide into 2 mononucleotides. In terms of the bond here, the name of it is called a phosphodiester bond. You need to know that name. So all those blue bonds here and then here are phosphodiester bonds. Then, if we've got lots of these polynucleotides, we can start to build up the structure of a DNA molecule. So, a DNA molecule is double-stranded. So, we've got a nucleotide here, joined by a phosphodiester bond to another nucleotide, and a phosphodiester bond to another nucleotide. Sorry about the drawing. And a phosphodiester bond to another nucleotide. And if I just go over those bonds in a different colour just to make it slightly clearer, you can see the upright shape of those nucleotides, just how I taught you to draw them. Now, on the other side, they join with the bases, with complementary base pairs, which we'll come to in a minute. But this time, these are going to be upside down nucleotides. So can you see that of the 5-sided shape, the point of those ones is always at the top? Yeah. The point of the ones on this side are going to be at the bottom. And the phosphate is therefore going to be just here. Right? So it joins with a base attached to the sugar, but on this side, we've got the point of the sugar going up and the phosphate down here because they're upside down. Like so. Oops, I didn't have a base attached. So we say that the 2 strands of DNA are anti-parallel because they're going in opposite directions, look. And then if we draw red bonds just so they match the other one, they'd be joined like this. Okay? So we've got 1 strand of DNA. We could draw an arrow to say it's kind of pointing in that direction. And 1 strand of the DNA going downwards, pointing in that direction. So we've got polynucleotide on this side, a polynucleotide on this side, and they're bound in the middle by complementary base pairing. A complementary base pairing in DNA is that A always binds to T and C always binds to G. Now, what they could do in an exam is tell you that these bases are these names, A, T, C, G, and ask you to fill these in. So the complementary one that would bind to A would be T. The complementary one that would bind to T would be A. The complementary base to bind to C would be G, and the complementary one for G would be C. So you could fill those in. The bonds in the middle here are hydrogen bonds. So the two bases joined by complementary bases join with hydrogen bonds. And A and T always have two hydrogen bonds. C and G always have three hydrogen bonds. So A and T would have two hydrogen bonds. So A and T again, two hydrogen bonds. C and G would have three hydrogen bonds. C and G, so that would have three hydrogen bonds. I always teach my classes T for two. I'll have tea for two, please. We need cups of tea for two people. And C and G both rhyme with three. So that's a way just to remember which ones go with which. Now, this structure here with the double-stranded is just for DNA. DNA is double-stranded. RNA is actually single-stranded. Okay, so DNA is double-stranded, RNAs are single-stranded. So you wouldn't need to go to this structure. The next part is how these then make this the kind of spiral structure. So we've said already that these are anti-parallel. A level of detail, that kind of the next step of that is labeling that. So you call this end the 5' end, and this would be called the 3' end. And this would be the 5', and this would be the 3' end. You might come across that in books. It's just a way to describe how they're anti-parallel, how they're going in opposite directions. Oops, I don't think that's how we spell parallel, sorry. So then these wind up to form the very famous double helix. Okay, so this strand is a polynucleotide, like we've learned to draw. This strand is a polynucleotide, like we've learned to draw. And in between those are those bases, A, T, and C and G, joined with the hydrogen bonds. Okay, so it's literally just this that's then twisted to form the double helix, which is the kind of final structure shape of our DNA molecule. Okay, and that's the end.